Okay, hello. Thank you all for joining us today. I'm Tracy Smith. I'm the Program Director of Education and Outreach for the Center for Integrated Cellular Analysis that's based here in New York City. And in this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we will be discussing new work that combines Perturb-seq with Parse combinatorial indexing and Ultima sequencing to systematically perturb signaling regulators across cell lines. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand, and I'll ask you to unmute yourself, or you can type your question into the chat, and I will read it aloud to the speaker. So our guests today are Carol Dalgarno, a research associate, and Longda Jiang, a postdoctoral researcher, both in the CICA lab at the New York Genome Center. Carol received her Bachelor of Arts in Molecular Biology and Biochemistry from Wesleyan University. Longda earned his PhD in Statistical Genetics from the University of Queensland, Australia, where he worked on developing computational methods for large-scale genome-wide association studies. Both Carol and Longda's current research focuses on learning signaling pathway signatures from pooled single-cell genetic screens in diverse biological contexts, and uses these signatures to understand signaling pathway activation for in vivo and in situ samples. So thank you both for being here today. Welcome. And Carol, I will hand it over to you.
Okay, thank you for the nice intro, Tracy. See my slides okay?
Yes.
Thanks. All right, hello everyone. I'm Carol and this is Longda, and today we'll be presenting on our recent preprint, Systematic Reconstruction of Molecular Pathway Signatures Using Massively Scalable Single-Cell Perturbation Screens. A central challenge in genomic data analysis is figuring out how to condense high-complexity omics data into interpretable biological conclusions. Gene set enrichment analysis is a popular approach that addresses this challenge by comparing lists of differentially expressed genes to a gene ontology or another similar database, and then returning enriched terms for cellular processes. While gene ontology is derived from diverse data sources, today we'll be discussing our approach, which uses a uniform assay, single-cell RNA-seq, to discover gene lists from perturbation experiments. This is a more systematic and data-driven approach to discover gene lists, which we call molecular pathway signatures that are involved in distinct cellular processes. Typically, single-cell RNA-seq is used to characterize cell type-specific functions in heterogeneous cell populations. However, it can also be used to multiplex together multiple treatments, which we call perturbations, in one single-cell RNA-seq experiment. Since these treatments can be combined in one experiment, it's highly scalable, and it also allows comparisons of multiple treatments with reduced batch effects. One approach which uses this multiplexed single-cell RNA-seq method is Perturb-seq, which combines CRISPR genetic perturbation screens with a single-cell RNA-seq readout. And Perturb-seq is very well suited to reconstructing gene programs by identifying correlated perturbations and then learning the associated gene expression profiles. One example of such a Perturb-seq dataset is genome-wide Perturb-seq, where they knock down around 10,000 genes in cells expressing CRISPR-I, and then generated an extensive resource of transcriptional signatures. However, this data set was generated in resting cells. So if we want to look at gene signatures that are context dependent, that involves in processes such as cell signaling, then we need to generate additional Perturb-seq data sets in cells that are stimulated to induce pathway activity. A well-established approach to characterizing pathway gene sets is through direct stimulation, where the pathway gene set is defined by comparing the transcriptomes of unstimulated and stimulated cells. In our study, we use a complementary approach by doing CRISPR-mediated genetic knockdowns of cell signaling regulators and transcription factors to learn the same or similar pathway gene sets. This has the added benefit of being able to dissect primary versus secondary pathway activation and being able to learn sub-pathway level transcriptional programs. With this type of data, we compare the non-targeting cells to the knockdown cells and identify causal relationships between a regulator or transcription factor and the genes that it regulates. And since we're performing Perturb-seq with multiplex perturbations, we can generate a large data set that we can use to identify correlated regulators and build pathway gene sets. In order to accomplish this, we decided to generate a large Perturb-seq data set where we perturb multiple signaling pathways and cell lines. However, since we're doing multiple perturbation screens, we need to profile a lot of cells in order to have enough coverage per perturbation. There are already many existing single-cell guide capture methods, but the majority of these use 10x, where it's very expensive and also labor-intensive, involving multiple reactions to profile large numbers of cells. Split-seq, which has now been commercialized by Parse Biosciences, offers a more cost-effective method for performing large single-cell RNA-seq experiments. They have kits where you can profile up to 1 million cells at lower cost using combinatorial indexing, and the method works on fixed cells or nuclei, so you can multiplex samples that you have prepared at different time points in one experiment. And this last point is especially well suited to our experimental design of looking at multiple pathway and cell line combinations. So we combine these two existing methods to develop a method to capture guide RNAs with the Parse whole transcriptome kit using guide-specific modifications. And before I go into what these modifications are, I just wanted to quickly acknowledge Effie Papalexi, a former PhD student in the lab, for her work on developing this guide capture method. So in the first step of the Parse kits, you perform reverse transcription with bar-coded primers. So in order to get guides alongside the transcriptome, we decided to use a crop-seq vector so that we have polyadenylated CRISPR guide RNAs that can be captured using the same primers that are already in the Parse kits. So from each cell, the guides and the mRNAs share a cell barcode. Then ligation proceeds as it normally would in the Parse kits, and 2 additional cell barcodes are added during 2 rounds of pooling and splitting. Then the next guide-specific modification comes in during cDNA amplification, where we add a guide additive primer that binds to the U6 promoter upstream of the guide, and this way we can enrich for the guides during the cDNA amplification PCR. And then finally, we do a guide-specific library preparation, where we do a PCR to add the Illumina adapter sequences, as well as a 4th cell barcode that doubles as a final sample index. So I just wanted to emphasize here that really only additions onto the kits that you would buy out of the box are adding these primers for the last 2 steps, which makes it easy to add on to things that are commercially available. So now that we have this method for doing Perturb-seq on a large scale, we designed an experiment to perturb multiple cell lines and signaling pathways. We used 6 different cell lines from different tissues of origin that all express CRISPR-I. And by performing Perturb-seq in multiple biological contexts, we're able to identify both conserved and heterogeneous transcriptional programs. We focused on 5 canonical signaling pathways and designed pathway-specific perturbations for each, where we chose genes to target based on a literature review so that we're perturbing known regulators. And this will give us a stronger signal that we can use to build the pathway gene signatures, since we're really focusing on the transcriptional profile of the pathway rather than identifying new regulators. To prepare these samples, we start with cells that are expressing CRISPR-I and then infect with lentivirus for 1 pathway guide RNA pool at a time. And then we simulate with the matching cytokine for that perturbed pathway for 24 hours before fixing and permeabilizing the cells to perform Perturb-seq. In total, this data set includes 30 biological samples, which cover 1,626 perturbations, and we profiled around 2.6 million cells, and we ended up sequencing with a combination of Ultima genomics and Illumina. And we decided to do this because a large part of the cost for these big single-cell RNA-seq experiments is generating the libraries, but another huge cost is sequencing, and Ultima is a potentially more affordable option compared to Illumina. So we sequenced all of our gene expression libraries with Ultima, and then also sequenced a subset with Illumina so that we can compare reproducibility of results across sequencing platforms. And then one final thing to note is that we had to sequence our guide libraries with Illumina because the fragments were too long for Ultima's single read platform. So here I'm showing just one example of one of the perturbation responses that we recover in this large data set. So here we have interferon gamma stimulated HAP1 cells that received an interferon gamma receptor 2 knockdown, and we can see that the differentially expressed genes include canonical interferon gamma response genes. We recovered over 1,600 of these perturbation responses, covering all 5 pathways and all 6 cell lines, and here I'm showing just 8 examples. So now I'll pass it off to Longda to discuss the computational framework used to derive pathway signatures from this Perturb-seq dataset.
Thank you, Carol. Let's dive in. So after obtaining our datasets, we soon noticed something very interesting. As demonstrated in this heat map, we noticed that the cells that receive the same guide RNA can have very different perturbation response. Some are strong, some are moderate, and some are weak. Previously, for the CRISPR knockout experiments, our team developed a method called mixed-gate to binarize the cells into perturbed and non-perturbed groups. However, these methods cannot account for the continuum nature in the CRISPR-I experiments as in this study. Therefore, we extended the original method into a new method called mixed-scale. Mixed-scale can quantitatively measure the perturbation strength, assign a score to each cell to reflect their perturbation response. To compute the mixed-scale score, we will first perform a standard DE test between the non-targeting control cells and the cells of the same guide RNA identity. And then we will aggregate the expression information from multiple large effect DE genes that we obtain from step 1 and convert them into a scalar and assign it to each cell. As demonstrated by these density plots here, the distribution of the mixed-scale score should reflect the perturbation response in each cell. To validate the accuracy of our mixed-scale score, we benchmark it against other known quantity in our datasets. So as demonstrated in these figures here, we can see that as the mixed-scale scores goes up, the expression level of the target gene goes down accordingly. This is consistent across different cell lines and different regulators, indicating that our mixed-scale score can indeed reflect the knockdown efficiency for the target gene. But you might wonder that why not simply use the target gene expression as an indicator to measure the perturbation response? Well, here we further show that our mixed-scale score is actually a more accurate measurement than the target gene expression. If you focus on all the cells that are located in the right-hand side blocks here, these cells are the cells that have no expression level for the target gene, JAK1. However, we can still see that there's some strong heterogeneity going on within these cells. But the mixed-scale score can correctly identify that and rank the cells based on that. So this indicates that the mixed-scale score, which aggregates information from multiple downstream regulated genes, is indeed a more accurate measurement than the target gene expression. We can further incorporate the mixed-scale score into our DE analysis. So instead of coding the control cells as 0 and the test cell as 1, now we will use the standardized mixed-scale score to code each cell to account for their perturbation heterogeneity. By doing so, we hope to achieve a higher statistical power and identify more DE genes. Tested in the simulations on the left, we show that our methods had a well-controlled false positive rate under the null, and tested in the real data analysis on the right, we further show that our methods can identify 30% more DE genes compared to the conventional 0-1 coded test. And more importantly, since we have 2 batches of data, it actually allows us to quantify the replication rate of the batch 1 significant DE genes in the batch 2 data. And we found that our replication rates is similar or even slightly higher compared to the conventional methods, indicating that our methods can not only identify more DE genes with higher statistical power, but also maintain a good robustness. As I just mentioned, and as Carol mentioned earlier in our talk, we actually have 2 different batches of data that sequenced by 2 different sequencing platforms, one from the Illumina and one from the Ultima Genomics. The ultimate goal is to merge these 2 batches of data and for a joint analysis to maximize the statistical power. However, before confidently doing that, we need to make sure that the data produced by the 2 sequencing platforms are consistent with each other. So we did 2 checks. The first check we did is to compare the CPN counts per gene. As we can see from these 2 figures, the CPN counts for each gene are highly consistent between the 2 sequencing platforms. And we can see that on a small subset of genes appear to be outliers. They're colored in the red. Most of these genes are non-coding RNA genes with a very low CPN count, and none of them appear to be significant signals in our downstream DE analysis in either the Illumina data or the Ultima data, so we can confidently ignore them. And the second check we did is to make sure that the DE results produced by the two sequencing platforms are consistent with each other. So we applied the mixed scale framework as well as the weighted DE test independently to the two subset of data. And we found that the DE results for each genes are highly consistent with each other. Given these findings, we confidently merged the two batches of data for a joint downstream analysis. So after merging these two batches of data for a joint analysis, in total, we obtained over 1,500 perturbations with at least 5 significant DE genes. So how can we utilize this massive scale of the results? There are mainly 3 aims we want to achieve here. First, we want to generalize our findings across different biological conditions for each pathways, and then extract the pathway conserved genes. And second, we want to compare our pathway genes with other existing pathway databases. And third, we want to utilize our pathway genes to interpret new biological contexts like the human diseases. So for aim 1, we first check the DE results for each pathways. For pathway like the interferon gamma, we found that its DE genes are usually highly consistent between different cell lines and different regulators. But for pathway like the insulin, its DE result is usually more heterogeneous. In this study, we want to emphasize that we mainly focus on the conserved genes across different biological conditions because our aim is to get a list of high confidence genes that we can utilize them to interpret other biological contexts. The cell line specific response is also very interesting, but that is not the main focus of our study here. So how can we extract the shared genes from each pathways from such multi-dimensional DE results? Well, we apply a newly developed multi-CCA-based method to do that. Multi-CCA is very good at extract the shared signals across multiple matrices and sequentially return you groups of correlated columns. We call this group the perturbation program, and each program associates a group of correlated regulators with a shared set of genes in both the positive and negative directions. We further call these genes the program signature for a pathway. To better illustrate what we're doing here, I'm going to give you one example for the interferon gamma pathway. So the interferon gamma program 1 that returned by multi-CCA covers all the core regulators in this pathway, as highlighted by the red circles on the left. We identify more than 600 genes as the program 1 signature, and most of these genes are highly consistent across different cell lines and different core regulators, as indicated by the different columns in each subgroups in this heat map here. These 600 genes form a very broad list of genes that cover the core pathway activity. But can we also identify a signature for pathway subbranch? Let's take a look at the program 2. So the program 2 returned by multi-CCA is positively linked to IRF1 and negatively linked to IRF2. We know that IRF1 is a downstream regulator of the interferon gamma pathway, and IRF2 negatively regulates IRF1. We identify around 200 genes as the program 2 signature, and most of these genes are a subset of the program 1 genes that we just identified. We call this program 2 the IRF1 dependent program. These results showcase that we have specifically delineated this subbranch from the core pathway. We apply this framework to all the other pathways in our datasets, and we obtain a bunch of different programs and their signatures. If you're interested in any of these pathways, please refer to our preprint for details. So now we have obtained our pathway gene list. The next thing we want to do is to validate their accuracy. So for each pathway, we collected a corresponding external datasets that we know the ground truth relevant pathway for validation. Take the interferon beta pathway, for example. We collected an interferon beta stimulated PBMC data that published in 2018 as a validation dataset. And we perform standard pathway enrichment tests for this interferon beta stimulated DE genes that we identified from this dataset, we find high enrichment for our interferon beta gene list, which is a good sign. But we also observe a high enrichment for the interferon gamma and TNF alpha gene list. All three pathways are highly correlated with each other, so this result is reasonable. But we soon realized that given the fact that our gene list contains a very large number of genes, it actually allows us to perform additional enrichment tests using these pathway-exclusive genes. The pathway-exclusive genes are the genes that are not shared between two correlated pathways. And by doing so, this time, we only observed that the genes are enriched for the interferon beta exclusive gene list, but not for the other two pathways. This indicates that the pathway gene list that we identify is indeed highly accurate. And we repeated the same analysis to all the other pathways that we have using different external data sets. So if you're interested in the details, please also refer to our preprints. All right, so the next thing we want to do is to benchmark our gene list against other external GeneStat databases. We chose the MCDB hallmark, one of the most widely used GeneStat databases out there, as a benchmark. And we observed a substantial overlap between our Perturb-seq gene list and the MCDB gene list, which is a good sign. However, we also observed there are many unique genes from both our ends and the MCDB ends. To better understand what are these unique genes, we went back to our Perturb-seq sequencing data and found that the MCDB unique genes seems to have a lower overall count compared to the shared genes and our Perturb-seq unique genes. Many factors could contribute to this. For example, the difference that the cell line that MCDB used and the cell lines that we used. So the MCDB usually use one or two cell lines to extract the pathway gene list, while in our study, we used a total of 6 different cell lines. The second factor could contribute to this is that the difference in the sequencing technologies between MCDB used and we used. MCDB usually use the Illumina-based sequencing technology, while in our data, we used a combination of Illumina and the Ultima Genomics platforms. We don't have a final answer here, but given the fact that the ultimate goal of this pathway gene list is to utilize them to interpret new biological contexts, we actually went back to our previous external dataset analysis and used them for additional validations. Take the interferon beta stimulated PBMC data, for example. This time, we perform a module score analysis using the shared genes, using our Perturb-seq unique genes, and the MCDB unique genes, respectively. And we found that the shared genes and our unique genes can well distinguish the interferon beta stimulated cells from the control cells consistently across different cell types. However, for the MCDB unique genes, it only performs well in some of the cell types, for example, the monocytes, but not in the other cell types. So this really highlights the comprehensiveness and the accuracy of the pathway genes that we learned from our multi-context strategy, while MCDB genes seem to suffer from certain limitations. So the next thing I would like to show you is how we validate the pathway subbranch signature that we identified. So we identified an interesting data set that collected samples with different kinds of defects in the interferon gamma signaling pathways. We tested for the expression level for the IF1 dependent genes, which are the genes that shared by interferon gamma program 1 and 2 that we just mentioned, and for the IF1 independent genes, which are the genes that are unique to interferon gamma program 1, respectively, in these samples. We found 2 samples show a very specific defect in the IF1 dependent genes, but not for the IF1 independent ones. It turns out that these 2 samples are the cells that collected from patients with an IF1 inborn deficiency. These results really conclude that the pathway subbranch signature that we identify is also highly accurate and reproducible, even using biological samples that is unrelated to our original Perturb-seq data. And in fact, we even replicated the same findings in an IF1 knockout cell line from the bats. This really highlights the generalizability of our pathway gene list that we learned from this multi-context Perturb-seq experiments. In the final few slides, I would like to show you how we utilize our pathway gene list to interpret new biological context. First, let's focus on COVID-19. We perform a very standard DE test between the COVID-19 patients and healthy controls PBMC using the Combat Consortium dataset. We then perform a standard pathway enrichment test for these COVID-19 DE genes using our pathway gene list. We find high enrichment for the interferon beta, TNF alpha, and interferon gamma pathways. This result is no surprise because all three pathways are highly correlated with each other, and they are all involved in the immune response. A more interesting question to ask here is which pathway is really involved here, while the other pathways are just showing enrichment because of their overlapping genes. To tackle this question, we perform additional enrichment tests using this pathway exclusive gene list as we just mentioned, and we found that this time, the COVID-19 DE genes are highly enriched for the shared genes, which is as expected, but also for the interferon beta exclusive gene list, and not for the other two pathways. This indicates that interferon beta might be more involved during the COVID-19 viral infection response in the human PBMC. These results highlight the importance of getting a comprehensive list of pathway genes as in our study. Otherwise, it is not possible to distinguish these highly correlated pathways. And finally, our pathway gene list can also be used to interpret spatial transcriptome data. For example, we identify a spatially specific TGF-beta activation in one of the regions in the mouse healing intestine. These regions nicely match to the inflammatory regions as annotated by the original study. If you're interested in the details of this spatial transcriptome analysis, please refer to our preprint. All the analysis and functions that we just mentioned are all configured into the Mixscale R package that is available at GitHub. You can use our package to analyze your own CRISPR Perturb-seq data and quantify the heterogeneity in your Perturb-seq data and also perform weighted DE test. Or alternatively, if you just have a list of genes of interest, you can also directly use our package to perform pathway enrichment tests using the standard pathway gene list or the pathway exclusive gene list that we just mentioned. So in summary, in this study, we first developed a new guide capture method for the Parse Biosciences whole transcriptome kit, and we then presented a highly scalable framework for multi-context CRISPR-I screens. This framework can be easily scaled up to screen millions of cells. And we also presented a computational framework to build a comprehensive and data-driven library for these pathway core and subcomponents genes. These pathway genes that we learned are highly generalizable to interpret other biological contexts. And with that, I hope we have convinced you that we are really moving towards the construction of a perturbation atlas in the near future. And that concludes our talk today. We would like to thank every members from the Strategia Lab for their help and support in this project. And thank you all for listening. Carol and I are ready to take questions.
That was great, Carol and Longda. Thank you both so much for sharing your work with us. So we'll move on to our Q&A section. If anyone would like to ask a question, you can raise your hand and I'll ask you to unmute, or you can type your question into the chat box. Let's start with a question about, and you just mentioned this discussing your GitHub, but what other kinds of perturbation data can you use Mixscale with?
So we mainly use our package to analyze CRISPR-I Perturb-seq data, but if there is a continuum nature in your data sets, you can also use our package to analyze your own data. For example, if you have your CRISPR activation, CRISPR-A data, you can also use our package to analyze that kind of data. And we don't have like restrictions for the types of like not CRISPR. So please feel free to use our package. Yeah.
Great, thank you. Another question is, so you started out in this preprint, you had profiled 30 biological samples, so you had 6 cell lines and various pathways. How much more scalable do you think it could be? How much bigger can you go? What are the limiting factors?
It's a great question. Yeah, so I feel like just from doing the hands-on work, obviously doing all the cell culture of these perturbations is quite intensive, but having the workflow where you can have pausing points of like preparing samples and then pulling them in an experiment, that definitely makes alleviates that strain. So I think with like the largest parse kit that we had, you could have 96 different samples in the plate for like all 96 wells and combine that many samples at a time. So if you have the resources to generate 96 samples, it could be scaled up a lot.
Incredible. And we have one more question here that just came in. How many cells are with non-targeting guides?
Yeah, so as like a standard for these Perturb-seq style experiments, we tend to have like 5 to 10% of our total number of cells will be non-targeting, so that we have like an adequate amount to compare to for statistical analyses.
Well, thank you both again we have a hand going up. I will ask you to unmute.
Hi, I'm unmuted. Hi, I'm Ritza. Thanks very much. This is a fantastic presentation and work. And my questions are very much from a signaler. So I noted that you've chosen a 24-hour time point, and presumably you also only had one concentration of each growth factor or stimulus, which I completely understand because the work that's involved in this is humongous. But I'm just wondering whether your signatures are capturing the chronic responses as opposed to the more acute responses to the signal. So because you're measuring after 24 hours, and so there will be a lot of negative feedback kicking in, and particularly in relation to your insulin and the lack of a consistent signature there. I do wonder whether some of that is because of the time point that's chosen and the concentration and differences in receptor expression as well. Just wondering what your thoughts are in terms of capturing the dynamics moving forward.
Yes, that's definitely a very good point. And we do see a very heterogeneous response in our insulin pathways. And we do hypothesize that that could be one of the reasons. So, but with our current work, we don't really have the capacity to really evaluate that. So maybe
Reach out to me if you want to.
Yeah. Yes, of course. We will definitely do that. We will reach out to you. Yes.
Thank you.
Thank you so much. Yeah.
Thank you so much for that. Longda, you had just mentioned this, but the question came up, could you clarify mixed scale uses? So you had mentioned works for CRISPR-I and A, but not for CRISPR knockouts.
Yes, thank you. That's a very good point. So for CRISPR knockout, we have already developed a method called mixscape because we think from our observation, the CRISPR knockout experiments the cells are usually more binarized. They form a very bimodal distributions, while in CRISPR-I or A, we see a more continuous distributions of the targeting cells. So that's the motivation why we developed these mixed scale methods to quantify that. So if you have your CRISPR knockout experiments, we would strongly recommend you try out our mixscape methods that we developed previously rather than the mixed scale.
Great. Loving all these questions. Can you speculate about why you have discrepancies between calling knockdown cells based on expression of the target gene or with mixed scale?
That's also a very good point. The main reason, I guess, is the expression level for a single gene is usually very noisy, especially in a single cell context. So the mixed scale, it aggregates the information from multiple downstream regulated genes, so it tends to be a more accurate measurements compared to the expression of just a single gene. That's we think why the main reason why we see mixed scale is a more robust measurements compared to the packaging expression.
Okay. And go ahead.
Hi, sorry, I have another comment actually adding on to the last question. Have you considered cell cycle heterogeneity when it comes to the response heterogeneity that you're observing? Because in the context of signaling, you will have some cells that respond very strongly because they're actively in the cell cycle. And then when it comes to the cells that are out of the cell cycle, they just won't respond at all, particularly for something like insulin. That's important. It could be related to why you're much better able to capture the responders with your mixed scale approach.
Thank you. That's a very good point. We haven't specifically quantified the cell cycle genes contributions in our data sets, but we do have previous data processing steps that we call the calculate Perturb-seq functions that we inherited from the original mixed game paper. During that step, that will regress out the cell cycle genes from the expression profiles of these genes. So we kind of think that we have already removed the contributions of or the impact of those cell cycle cycling genes. But for insulin pathway, that I think that is not adequate. So that's a very good point. We might
and
look into
apoptotic
that.
cells or signatures, if you
Yes.
can. I know that a lot of those are filtered out when they have low counts and things, but it's just worthwhile just having a look. It's something we see a lot when we profile signaling.
Yes, thank you so much. That's a very good point. And we will definitely reach out to you for more discussion. Thank you.
Well, great. Thank you all. Well, thank you, Longda and Carol, for joining us today, and thank the community for this lively and lovely discussion. Hope it continues moving forward. So in closing, this seminar has been recorded and it will be available soon on YouTube and on our website, which is multimodalintegration.org. You can also follow us on Twitter at segs_ICA, or check our website for the latest announcements regarding upcoming talks and events. With that, I'd like to thank everyone for joining, and we hope to see you at the next seminar. Have a lovely day.